Good morning, everyone. Here we are at week number 8, the last week we have together in Biology 120 lab. So last week, what did we learn about? Carrying capacity of ecosystems, how many individuals of a species can an ecosystem support? You also learned about cohorts and studying cohorts and populations, and that maybe you chose males and females who were born between 1770 and 1810 through the gravestone cards that you used. Those are very interesting, and those are ways that ecologists study populations, which we're going to do again this week. This week's topic is all about measuring population sizes. So the best way to measure population size is to count all of the individuals in a population. Can you normally do that? No, you cannot, because you don't have unlimited resources no matter what your job is. And so usually ecologists who study population size sample populations, and that's what you're going to simulate today, this week, with your lab. You are going to use transects. So these are lines that you draw through this ecosystem, your board with all the blue, red, and green squares on it. You're going to draw lines of certain length across this ecosystem that you have, and you're going to count which individuals, how many individuals, either red, blue, or green, you have at a particular intersection. Well, you're going to count which ones you have at a particular intersection with the line. So this stop, you see a red one. Next stop, you see a blue one. Next stop, you see a red one. And you're going to write those down in your data table. So those are transects, and you can get an idea of your population size by knowing how many individuals of each type you have overall in all the sampling that you do with these transects. You can also use quadrats, which are basically a measure of how many individuals of a species you have in a particular area that has been marked off in a grid sort of fashion. So usually if you're an ecologist, you're going to measure 1 square meter and then count how many plants you have, for example, in this quadrat. Maybe it's an endangered sunflower, like we have, wild sunflower, like we have in South Carolina. So that's an application for a quadrat. Of course, if you're measuring something like bird populations, you're not going to use transects or quadrats, because if you walk into an area, all the birds fly away, and so pretty hard to count them. So instead, you might use the mark and recapture type of population sampling. This is very important for something like bird populations. I know down at the beach, oyster catchers, I think, are on the threatened list. They're so interesting. The black and white birds with a bright orange beak, and they love to open oyster shells and eat oysters. And so there's a famous oyster catcher down at the beach where I go whose name is U2, and that's because he has a little wristband on his little leg band, I guess it is, and it says U2 on it. And so everybody who sees him reports where he is, where he's located. He has a mate, and they have successfully raised chicks for quite a few years now in a particular area right where I go to the beach. So that's mark and recapture. So you're going to try that simulation with some beads, with white beads and red beads. Okay, resources for the answers to the examples this week. Go to your OpenStax textbook, chapter 45, section 1. It's all about ecology, and that section 1 is all about measuring population size. The examples are there, and the explanations are there. Now, there is not an explanation for transects and why you would use transects, but you can figure it out because that's very similar to quadrats. All right, hints for the lab. It tells you to use a wet erase marker. You don't have to do that. Use your wax pencil. You already have it in your kit. You don't have to go look for a wet erase marker for this. The wax pencil wipes out just fine with a paper towel. Also, the main thing for you to get out of this is why you would use a particular population sampling technique and why you would not use this technique. And I already gave you an idea. If you're counting birds, you can't really use transects or quadrats. That is just not going to work. And I know because I'm a birder, and yeah, as soon as you walk into an area, they fly off, especially the really skittish ones. So, why are you going to use this particular population sampling technique, and why would you not use it? And that is important, and that is discussed in your OpenStax textbook, chapter 45, section 1. So that's your lab for this week. A couple of reminders and comments for you. Please make sure you finish any unfinished work this week. Everything has to be turned in by Sunday night, June 26th, by 11:59 p.m. If you try to submit something at midnight, it will not go in the course. And you can't send me something to put in because I can't either. All I can do is grade at that point forward. So make sure you get everything in. If you have completed 70% of your work or more, and you're still having a tough time finishing everything, then you can let me know and we can request that you get an incomplete for now, and then you have a month to get everything completed. But you have to have 70% of your work finished, and you only have a month extra if it is approved. And that is not up to me, that is up to the registrar. So just so you know, you do have that option, just in case things are happening and you can't get stuff done. The other thing that I would ask you to do is please fill out your course evaluation, because this helps me as an instructor be a better instructor. You give me hints, you give me critique. I am fine with that. I will take that to heart and improve my teaching. So please do your course evaluation. It also helps my team lead, who is over a whole group of faculty members, judge how I'm doing based on your evaluations. So I ask you to please do that for me. That would be greatly appreciated. And if you need anything, there's my email address. It's been there all semester long. Let me know if you need anything. m.sigmon@snhu.edu. It has been my absolute pleasure to be your instructor for this lab. We've learned a lot. We've done a lot. Just think pea experiment, and also the fast plant experiment. Yeah, so we've done a lot. I hope you've learned a lot of biology, because it is a wonderful, wonderful subject. And I wish you well in the future. Let me know if you need anything, and keep in touch. I love to hear from former students. Have a wonderful week.